The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 17th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the weekly bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam Cotty, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian Wile, and I've been in the CRO industry with flow cytometry focus for about 13 years.
And you know, Brian, I was thinking that it's a new year, so we may need to update those numbers, but I'm not getting off topic.
We count by twos or threes, I forget. Yeah.
Right. Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with additional locations in Europe and a partner location in Australia, we're both here in Philadelphia, and Jeremy, our producer, is in Missouri, so we're truly all over the map. We're overjoyed to have you listening to the 17th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing, before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're exhilarated to have you here, and we're looking forward to a fun episode today. To kick us off for episode 17, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. So we have another immunology-packed agenda for today. The first one is a little bit of a trick because it's a news article that's relatively recent, but it keeps referencing a study that is a little bit old. But it's around a peanut allergy trial that's currently running in Australia.
Yeah, this was really interesting because, peanut allergy is obviously something that can be very dangerous. As you know, as somebody who has that sort of allergy, it could be as bad as the airway closing up, going to anaphylaxis. And so want to make sure that you can prevent that or fix it as quickly as possible so there's no damage. And with this, what we're looking at is actually two IgG4 antibodies that block IgE-mediated effector cell activation. So really, it's blocking the effects of the allergy there. These are competitive antibodies. Now, the thing that I found really interesting, as you might imagine, how do you test something like this, right? You have to administer peanuts and then have some sort of attack and then administer the treatment. And then you also want to see if later on it prevents another attack later on. And so they, you might imagine, had difficulty recruiting subjects for this. It was a placebo-controlled trial as well. So there were a lot of hurdles there to make sure that it was done properly. It got me to looking up some of the NIH clinical center principles. So they have 7 main principles for conducting ethical research and, spurred it on. So one of them is social and clinical value, scientific validity, fair subject selection, favorable risk-benefit ratio, independent review, informed consent, and respect for potential and enrolled subjects. And so a lot of it there, these studies have to meet these kind of criteria. And now I will say this study specifically is outside the US. This is Australia-based, if I remember correctly. But still, I mean, these kind of guidelines are fairly universal there. But did you find out as to where this study is at and the results?
So they have not released interim results yet. They are still enrolling subjects. And as you say, they are definitely, I think one of the reasons that these news articles keep popping up every 2 or 3 months is that likely clinical sites are trying to advertise and find people. you mentioned the cost benefit analysis, and you always have to take that into account with patients. The cost to them is some severe discomfort and a lot of worry, and being put into a relatively dangerous situation. But you think about the benefit too, right? Where if they go into the wrong restaurant and there's an accident and a peanut gets into their food and they go into anaphylactic shock and no one has an EpiPen or knows what to do, these people could die on a daily basis. So I can see definitely a benefit to it. But yeah, it is a daunting ask for clinical trial subjects there.
Well, I even had a story like that. Went to a minor league baseball game and had bought a bag of peanuts. This is something I usually did with the kids at baseball games, anyways. And we go to open it up, and several seats down, a woman came out. She's like, "No, no, no, you can't open that. You can't open it." And keep in mind, I mean, we're outside stadium. And just told me that her child had a peanut allergy, and so we didn't open up the bag of peanuts. But yeah, I didn't even think of it as being like that impactful. I guess if you had a peanut allergy and you had one of those little skins fall on you or something, like it blown over, maybe it could cause some sort of reaction, or you inhale it or something. So to your point, yeah, that cost benefit analysis in that case, I would imagine that the benefit would far outweigh the cost there.
That's it. But definitely an interesting thing. And again, definitely immunology focus, where we're really looking at this outsized reaction in IgE and likely some basophil interaction there. So one of my favorite tests is always the basophil activation test as well. So I'm sure that will be implemented in this trial, and wishing the trial participants and study sponsors the best there as they move forward.
For sure.
All right. And in terms of the next story I had brought up, that is around a paper that actually got accepted into Nature Immunology. So here, this is actually in New Jersey, a little bit closer to home than Australia. So just down the street from our Philadelphia lab.
A little bit.
Right? Dr. Zhu is basically modulating the immune system to fight diseases in an entirely different way than has been done before.
Yeah, it looks like what they're doing is finding a way to transition effector memory T cells over to central memory T cells quicker to enable a more rapid response. And so, as listeners likely know, T cells are part of the adaptive immune system, and central memory, as it might indicate, the T cells learn on exposure to an antigen, and then they can respond much faster than a naive T cell, one that has not yet been exposed to that antigen. And so here they're looking at TLE3, a transducin-like enhancer, which is a transcription cofactor, and it functions in training T cell training. And one of the papers I had found was actually that by ablating this gene, this protein, that it then actually sped it up. So it was interesting there.
Yeah, and with any of these types of papers that are finding sort of a new mechanism for immune regulation, I always like to think back to sort of the history of PCR application. And as part of that, scientists first found one or two enzymes that could replicate DNA, and everybody was very excited. But it was still sort of an expensive process because you had to have very carefully controlled buffers and very carefully controlled heat sources that would cycle up and down and do different things. And then throughout the years, people kept discovering more and more enzymes that could do different types of DNA replication, have slightly different properties in them. And, at some point you look at literature and you're like, oh, do we need yet another enzyme that can do this? But then you look at what sort of niche applications you can find, and, maybe you need one that can operate in a high salt environment because the output of your previous step is high salt. And then maybe you need another that is point of care, so you can't have a thermal cycler there, but you can have something for a controlled incubation for 20 minutes. And then even though it's maybe not as high fidelity, then it's still functional in that context. So when I see these different types of T cell regulation techniques, I always think about, great, now we're expanding our tool set of what we can do to immune cells to affect the response that we need in a more controlled fashion.
When you were starting with that, I thought you were going into the whole light bulb driven heat sources and dipping it in water baths to do your PCR. But no, you weren't going that way. You were going really old school. Will say that the first lab that I worked in out of college was an academic lab, and the PCR machine was actually heated up by light bulbs, which is kind of crazy.
An easy bake PCR oven.
Yeah.
Yeah, but I did find this article nice, and again, I like any article that refers to a journal article. This makes me feel much happier and much safer in reading the article and trusting some of the outcomes. It's always interesting to see how the medical literature can make it into popular media there as well.
Yeah, agreed.
All right, and then switching over to investment news. The two stories that I brought up today are around Barinthus Bio. So it's yet another of these companies that is switching its strategic focus over to the immunology and inflammation area.
And this platform was really cool. So we're talking about the lead molecule here, VTP 1000, specifically for celiac disease. It's a phase 1 study. The platform is SNAP TI. So essentially combining multiple antigens and an immunomodulator into nanoparticles. And what they do is then target it towards the antigen presenting cells and draining lymph nodes, spleen, and liver. Essentially, what they're looking to do for this is to optimize the Treg to T effector ratio, trying to reduce inflammation, as you might imagine, in such an autoimmunity here. These peptides are amphiphiles, so they're hydrophilic and lipophilic, and are actually self-assembling, which I found really cool. It then gets injected intramuscularly and reduces the size of the particles that reduce the systemic effects here. Definitely all that technology stuff was really positive. Now, I will say, as far as the investment side of it, the company is reducing 65% of their workforce to extend the runway through the beginning of 2027. This includes the chief operating officer, chief financial officer in the UK site that are leaving as well. And so they're looking to partner some of their other assets, including the VTP 300, which is for chronic hepatitis B currently. So there's your investment portion for it.
That's it. And, we have definitely seen a decrease in the spend on infectious disease. So that's no surprise. Basically, it was a very hot area to be in and was likely great for fundraising for quite some time. And now the funding has moved, and this company is pivoting with the funding over to the immunology research side. So definitely on the funding side, I think it's a common story that we're hearing about with companies renaming, consolidating, paring down their pipelines, and really focusing in on what they think are the highest value assets there. Mm-hmm. In terms of a treatment for celiac, it's become a lot easier to go gluten-free overall, but definitely I'm sure there are plenty of people that would prefer to have that immune response turned off, and especially in like severe allergic situations, it would likely be required for living comfortably. A nanoparticle injection does seem, on the harsher side, I guess, for celiac disease.
Depending on how often, right? The frequency of administration is certainly one consideration there. But, and obviously this is taking the lighthearted aspect of it. I mean, being able to have regular pizza instead of gluten-free pizza is certainly one that I'd prefer. I don't know for how many.
You're saying your cost-benefit analysis says yes due to pizza availability. So yeah, I mean it's a valid concern, right? So
obviously for me, I may be an N of one though, and I don't mean to make light of it. It certainly is a very a disease that certainly needs the support, and so hoping for the best there.
That's it. And with a lot of these formulations, sometimes they'll start off with an injection, and then when they see efficacy, then they may look at different formulations that may be more tolerable or have follow-on developments or formulations that can increase patient uptake, so that the severe people can basically take the medication as soon as possible with an easy-to-make version that's injected intramuscularly. And then they'll have the cash runway then to develop the medication for easier access for patients.
Right. Yeah. Yeah. So very interested to see how that plays out.
Yeah. And then the last investment news is one that we didn't cover last year, so I wanted to bring it up again. So AbbVie, a company that we always watch, especially in the immunology space, acquired Nimble Therapeutics back in mid-December.
And so this is another one with a focus on autoimmunity. They're looking at the lead asset is oral peptide, IL-23 receptor inhibitor. Specifically here, the indication is psoriasis. And interestingly, with oral, it's one of those ones we really have to work on modifying so it has stability in the gut. It just doesn't get broken down as it's ingested. And so it also has extended half-life. The thing that I thought was cool was that it was generated, the therapeutics generated using light-directed peptide synthesis platform. And this is something that could generate over 100 million peptidomimetic compounds overnight. Really hadn't looked into much of peptide synthesis in this way, and specifically not light-directed, and only really did very brief look into. How familiar are you with that?
With that specific manufacturing pathway, not. But I will say that the CDMO and drug manufacturing world in general has been exploding in terms of creativity and possibility as much or more than the CRO and analysis world too. So definitely the number of techniques they have and the amount of control they have over these antibodies is very impressive now versus what it was. You look over the history of antibody development, and it's roughly 50 to 60 years at this point. And the first antibodies, weren't really well controlled, right? It's very similar to how we view cell therapies and gene therapies today, where it was basically at the edge of their technological capabilities at the time to produce these and characterize them, and it wasn't as clear how exactly you needed to characterize them to make sure they were effective and safe. But now the amount that's known about how to make an antibody, how to make it safely, how to make sure it's effective, and how to assess it afterwards. I know Dominic and company in our sister podcast there at the Weekly Bioanalysis talk quite a bit about how hybrid mass spec and LBA have really revolutionized that field and allowed us to understand that. So
we've got scientists here who have had vast experience in antibody generation. Some of the prior stops in their career. Myself, I was over a discovery group doing very much that same thing. And, you learn about the different substitutions that you could make to increase half-life and alter the modifications just to make it so that it becomes an effective therapeutic.
That's it. Yeah, so definitely exciting times. Nimble is definitely at the front of the pack, so we'll be excited to see what capabilities this adds to AbbVie and how they move forward here. As we talked about last time, there are quite a few companies, including AbbVie, that are coming up on relatively significant patent cliffs. So this makes sense as another one of those sort of mid-sized acquisitions, mid-sized to smallish on the big pharma scale acquisitions. To really shore up their pipeline for the future. So they're looking 5 to 10 years out as opposed to like next year.
And I did notice really sneaky there. It wasn't in any of the agenda notes or anything like that, but you got cell therapy mentioned in there because I think we need to have it mentioned at least once every single episode, right?
Oh, yeah, of course.
That was a
cell
good
therapy
job.
and puns. That's what we do here.
So what do we got next?
Next up, we've got some KCAS news. The thing that I wanted to highlight this time is that KCAS is joining the Mid-Atlantic Flow Cytometry Association as participating members there.
Yeah, so let's talk about this. It's another flow association. Thinking MetroFlow is a type of one. There's some others. Tell me how this one's different.
Yeah, and it's very similar to these other types of organizations. So you mentioned MetroFlow. There's also Glypha and Flowtex and NorCal and a couple others. And essentially, these are all sort of mini CITOs, if you will. And CITO is, of course, the biggest international flow conference, but it's only held once a year, and it's potentially expensive depending on your budget. So most groups will either some may not be able to go, some may only be able to send one or two people, and those are relatively senior people. So then, if you're a junior flow cytometrist, your options for being exposed to new vendors and sort of interacting with the flow community writ large are limited. So the thought behind this group is to get a Philadelphia and sort of mid-Atlantic centric, so anywhere from the Philadelphia to DC area group that can meet on a regular basis throughout the year, discuss things like CITO, but then have a local networking group. That way, junior scientists can have a place to go and, again, network with their peers, but then also practice their presentation skills and learn about new developments there.
Yeah, I mean, it's really important to make those connections, right? I mean, we all know that a part of being able to find a job is to make those connections and also advance your career, learning about new technologies, processes, things of that nature. And so really giving the people in this area that opportunity.
That's it. And as you say, there are all these other communities in these other areas, but there's been nothing sort of formal in the Philadelphia area for a while. So hopefully this will address the need, and people will be interested to join up.
That sounds really cool. So what's next steps there?
Next steps for MAFCA or next steps for the podcast?
Next steps for MAFCA.
Yeah. So for MAFCA, we've organized quite a bit of the committees and got some goals for the year. So we're tentatively looking at a meeting on the 12th of June. That'll be open to anyone interested in the Philadelphia area. So if you want to talk about flow cytometry, we're going to be summarizing CITO. And talking about some original research and just networking over some appetizers on the UPenn campus. So we'll make sure to put a link to that on the podcast as well.
And you mentioned the Philadelphia area just because this is a shorter duration, just a single day meetup rather than more of an extended thing. Otherwise, you would be inviting from the entire region, essentially.
That's exactly it. This is like an afternoon meetup on a Thursday afternoon. So anytime between 3 and 6 is basically the formal meeting where people can stop by after work or whatnot as they're able.
So where can I get my T-shirt?
I don't know if we have T-shirts up on the website as of yet.
Fair enough. I know you mentioned it just really started, so putting that out there as a thought.
Yeah.
Excellent. So, all right. What else do you got?
Next up is the FDA biomarker validation guidance that came out in January.
And so we don't have an impact factor for this, right? This isn't something that was in a publication. This is, brought forth by the FDA. And so looking at what are, I shouldn't say requirements, right? Because as it says, it's guidance document, how it's recommended to proceed. And so here our primary interest is flow cytometry, and the guidance that we use is the CLSI H62, specifically as it relates to validations. And so it's important then to understand if there is any contraindication, anything, that might be different here. And so what have you found?
Yeah, I think there were a couple interesting things in what you said, right? That you're absolutely right. First of all, this is not a paper. We are going off script a little bit to review a thing that came out. And it's very interesting to think about an impact factor for FDA guidance documents. I'm gonna propose that to somebody at WRIB, I think, this year. Yeah, I mean, the FDA is the organization that controls the drug development process, right? So they say what data is admissible, what data is strong enough to be considered for decision making, either in study to determine like what dose level a patient should get, or to make decisions on if a drug is effective or not effective. So definitely, what the FDA says is the most important. As you say, we do follow the H62 guidance documents. These are not actually FDA endorsed documents, right? So these are CLSI, so the Clinical Laboratory Standard Institute document. So it's a similar organization, but it is not a formal guidance for the drug development industry.
But it is produced by subject matter experts within the industry. And so it's not just a document somebody threw out there. A lot of authors on there, a lot of information that's very pertinent and really should be understood if you're in that area.
Oh, 100%, right? And I think that's the next thing, that really this biomarker guidance primarily just pointed back to another document called MCH10. And some of the confusing parts about that pathway is that it pointed back to it, but that document specifically says that it's not applicable to biomarker validation.
Right. And no, I think the one thing that you would pointed out was that the European Bioanalysis Forum had actually responded to this and were specifically calling out that the ICH-M10 mentions these biomarker assays are out of scope, that it focuses on PK assays, right? And so really what they're looking at here is to try to understand exactly how this decision came about, how this guidance came about, feeling like there wasn't as much of an attempt to collaborate across the industry, right? And what they're advocating for is more of a context of use.
That's right. So, and in terms of the context of use argument, it's essentially, again, the fit for purpose type explanation where we are saying, is the evidence for this biomarker strong enough, and do you have the clinical data to back it up? And understanding that each biomarker, will be used slightly differently, that means the data behind it may need to be different, right? Mm-hmm. Which makes a lot of sense, but makes it very difficult to regulate or to really pin down in a certain area.
It does, absolutely. And, we've talked about fit for purpose plenty of times because by and large, that's what we do, where it's, we make assays, we do panels that are fit for purpose based on client needs, what kind of markers they're interrogating, what kind of receptors their therapeutic is targeting. And then two, as we go through the various levels between qualifications and validations, and making sure that it addresses what the endpoints are for the study. And so the vast majority of the work we do is secondary endpoints, non-decision making endpoints, and exploratory endpoints. And so understanding how you would generate a validation or qualification that would suit that.
That's it. And, I agree with you again that H62 was put out by experts in the field, is current best practice, and is the most comprehensive document to date, right? And I think it addresses a lot of the things that flow cytometry needs that don't get addressed in other areas, because those problems have been solved in those other areas, right? So H62 addresses instrument setup requirements, compensation requirements, different ways to combine different patient sets, because it's known how expensive and difficult some of these samples are to get. So it goes a lot farther than some of the other biomarker guidance, and is very specific to flow, which I think is required.
Yeah, you need to make the most out of each of those samples that you're getting. They're very precious, and as we know, I mean, they're supporting clinical trials. So you have patients that are ill and still uh, providing that to be able to benefit the study itself and other patients down the road.
That's right.
And then, so I mean, along you got the lines here mention about H62. There's more documentation for that came out just last year. And looking at recommendations here, or further recommendations, when you're making changes, what kind of parameters should you evaluate with those modifications? You have to keep in mind, what did you do? How should you look at it? And then they had case studies around that. So what am I talking about? If you've made an instrument change or a platform change, you've substituted antibodies, added antibodies, things of that nature. And so really understanding, well, I did this. How should I make sure that I've still got a validated assay?
That's it, right? So yeah, those were the two big changes. Change one was, if I modify my assay, what's required to requalify it? And then change two, and I call them changes. They're really just addendums, right? That weren't covered in the initial, because the initial was long enough, and you got to cut the scope off somewhere. But so these addendums. So addendum one was the change to the assay, and then addendum two was how to work with rare matrices that you may not have access to during the validation.
Right, and that's always something that we struggle, and I would say struggle with, because we've gotten solutions, right? You want to try to get disease state samples if you possibly can. If you can't, then other options are stimulated healthy donor blood, just straight healthy donor blood. You could also get healthy donor blood that's spiked with either cell line or other sample that recapitulates that disease state. And so certainly there are workarounds there, but then making sure that it's something that is as close to the disease state sample that you would be working with.
That's right, or guidance for how to use in-study samples for additional validations as well. So, definitely covering those whole things. And that's the nice thing about H62 is that, again, it is very flow focused. So it was a very comprehensive document when it came out. It's a living document, so it's constantly being added to.
Mhm.
But, it does always leave that gap that would be nice to fill of, what does the FDA need to see for a flow cytometry assay to be validated? And that does not have the same level of clear guidance, and has a lot more variability than other fields like LBA and mass spec.
Well, I think too, there is some distinction here, right? There had been more recent guidance on, was it laboratory derived tests? And when you're looking at things that serve a primary endpoint on a clinical trial, there is more governance around it. I think CAP-CLIA assays. And so I see that as evolving ahead of the pack, right? And kind of driving, pushing that conversation forward, which will then filter down to a lot of the secondary endpoint and exploratory endpoint assays that we do.
Right. And, I would think that would be the case. I just think about, what would perfect look like, right? Like, what would a well-regulated flow cytometry system be? And, one of the things I think would help the field out the most is if there was a standardized electronic report format.
Mm-hmm.
I know there's quite a bit of effort going on with CDISC and others to make standard naming conventions
Right.
for the output. But I think a standardized report format, again, like MCH10, where, if you say table 1 of MCH10, people know exactly what you're talking about, what information to expect in what format.
Mm-hmm.
Whereas I don't think that the same thing exists in the flow cytometry world, where there are defined reports. Everybody has their own report. Like, we have our standard format, our sponsors have their standard formats, and we all try to, adjust and meet, in some sort of middle ground. But if there was an industry standard reporting format, I think it would be a huge benefit to the field. Because everybody would, stop using that as a differentiator, which I don't think is helpful, and B, because it would make all these reports machine readable, and then contribute to what we could get out of AI overall.
Now, talk to me about where you see that fitting, because when I think about reports, I'm thinking about a lot of different reports. We've got the validation qualification reports. We've got the bioanalysis report. We have a bioanalytical report. We've got sample analysis report. And so I'm imagining that what you're talking about here is the sample analysis report, but let me know what your thoughts are.
Yeah, I'm thinking of the validation report and the bioanalytical report as the two things that the FDA would be most concerned with.
Gotcha. Okay.
And yeah, I think that a standardized report format would be helpful to the field. And again, enable this sort of cross-trial and cross-platform comparison between different things, especially if it starts to incorporate things like CDISC. That's then becomes like a ratchet to move the whole field forward. So that way people can stop saying Tregs if they mean, specifically CD27 or CD127, and maybe we can define Tregs as only FOXP3. But until there is some sort of centralized guidance and real requirement from a regulatory agency, I don't think the field can harmonize or will harmonize quickly enough without that.
Well, it makes it hard to really mine the data sets too, right? All the data that's out there, it's essentially in all slightly different languages, if you will.
That's it. Yeah.
So what you're telling me is when I selected the type of report you were talking about, I was totally wrong. I picked the only one that you didn't pick there.
I mean, it's a subfield of the report, right? It gets included in the bioanalytical report. But yeah, I think the FDA generally won't look into like single samples for the submission level entries, right? So.
Right. And I think the way I was going with was the data that's generated by the clinical trials, right? So you have your sample analysis report that does the true information is actually in the cumulative files, the output data there. See the direction that you went there.
Yeah. All right. Anything else that we wanted to talk about with validations or regulations on validations for today?
I just think as far as validations and qualifications are concerned, it really is one of those things where you have to understand your assay to piece together which test scripts you think are most important. You have to understand your budget, right? Because I think everybody comes in wanting to have every single test script and, put together a gigantic qualification validation, but you can still get what you need without having to run every single one of a test script. And so that conversation is important. You bring that to our team. Our BD reps are very knowledgeable in kind of walking you through the options, and then our scientific advisors really have that true in-depth understanding to help fine-tune it for you.
Yeah, exactly. All right, and maybe as we roll out, I'll give a lighthearted fan feedback request and then I guess two lighthearted fan feedback requests. So the first one is, and I know we'll have some diehard fans that feel very strongly about this. I'm anticipating some good feedback here, but what type of font do you prefer for your flow cytometry reports?
Oh, no.
I know.
Wait, I know yours has to be Comic Sans, right? No?
Of course, wingdings, right?
Wingdings.
No, but
all right, maybe not. Hey, I will say though, when I was in college, I used to love that, and then everyone's looking at me like I got two heads. I'm like, I gotta stop using this. It's not really appropriate anymore.
That's it. But do we need like a strong like Times New Roman or a very sort of matter of fact like Arial? Like, what is the font that we need for reports? So yes. I'd be interested to hear. And then the second one is you mentioned test scripts, and that's what at KCAS and the Flow Cytometry group, that's what we call each subset of validation criteria, right? And the second feedback request is, what do you call a test script or a validation criteria? I've heard some people call it test scripts. I've heard some people call it a precision. I've heard some people call it a validation criteria. But I'm interested in what different people call these different validation steps.
Well, I've
So,
seen individual ones be precision. You have your inter-instrument, inter-analyst precision, but then you also have robustness, you have repeatability. So I always thought, I guess, precision is being a little bit different. Yeah, by and large, I've heard test script, but that could probably be just, where we are. That's what we always talk about.
right,
We'll
it's
be,
a pop versus soda issue.
yeah, it really, we'll be curious to see what comes out of that question. It's a good one.
Yeah. All right. Excellent. Well, looking forward to some good feedback there from our fans. And then I guess we'll wrap up with some banter. So we've each done a little bit of travel lately. So some trains, planes, and automobiles. But I want to just bring up the complicating factor of snow.
Complicating?
Yeah,
Complicating? Come on.
complicating. My feelings on snow are strong and negative. And just wanted to get your take, Adam, on which mode of transportation is most reliable when it comes to these winter weather storms.
Me and my car.
Yeah.
No, it's interesting because maybe I'm just like a unique person, maybe, and that's not said in any way that I'm trying to elevate myself. It's almost thinking that I'm on my own island here. Talking about travel, I went up to New York City for a client visit just yesterday, Wednesday and Thursday. So I got to drive in New York City in Manhattan, and I grew up in Queens, right? So I got to be honest, I loved it. Like I felt so comfortable. It just felt right to me. And I tell everybody that I tell this to, they look at me like, "What is wrong with you? You like driving in New York City?" I'm like, "Yeah,
And on top of that, in the snow,
in the
like
snow."
was there snow there at the time or no?
It was on the sides. It really
Okay.
wasn't much all over the place, but I don't know. I guess sometimes you grow up with something, no matter what it is, you get used to it, and then you come back to it, you like it. Maybe that's it.
Okay. So we need to have like a fast moving yellow car coming at you from the side at most times, and then you'll feel more comfortable. You're saying.
Something like that. Now, I will say though, you were asking about which one's the most reliable in snow. The day that we were going up, it was snowing that morning, and so there was questions about the plane for one of the BD reps and whether or not her plane would be canceled. One of the BD or the SSA was going on train, and she wasn't sure if her train was going to be canceled. All I had to do was look at the roads and see what traffic was like, and then I got Waze to tell me which way to go, or whatever app you prefer to use there. And I had no problem. In fact, I got there nice and smooth, no delays whatsoever.
Okay. So what I'm hearing is cars are the way to go.
I am sure that compared to the other questions you just asked, if you put something like that out there for our fans, we're going to get a lot more fiery feedback.
Okay, excellent. Well, yeah, we'll see what people say there as well. But excellent. Well, glad that you made it there safe and sound, and were able to drive around with all the crazy New York drivers and get your New York driving fix. But that's it for our show today. Next time in March, actually not February, we're looking at ideally instrument harmonization and what's in an MFI.
Yeah, so looking forward to that. Should be a very good conversation. You really, I know you have a phobia about your snow and whatnot, but you didn't say which is your preferred mode of transit when it's snowing out.
I tend to just want to hibernate as much as possible, right? But I would say if I had to pick one, I'd pick train for snow overall.
Makes sense. Yeah.
Yeah,
I mean, it's on the rails, right? So technically it should be fine.
that's it.
I just remember again growing up in New York City and the subways, there's lots of snow, you'd have delays and all of that. But I mean, if the subway's getting delayed, if the buses are getting delayed, then everything's getting delayed. So it's a mess.
Goodness.
Sorry, we can move off of this. We can think about sunny weather. What is it down there by you now?
It's like a lovely 50 degrees.
Here it says that we're getting 3 inches of snow Thursday. So
Yep.
sorry, I meant to get off of that topic. All right. Well, I think we should wrap up here. It's definitely been a fun episode, and really appreciate the conversation, and looking forward to the next one. Thanking all of our listeners out there. Looking forward to hearing any feedback that you have, and thanks, everyone.